Hi everyone, I'm Alice Merkel. I'm a collections assistant at the Maryland Archaeological Conservation Lab, and welcome to STEAM Sunday. We're here with Justine McKnight to talk about archaeobotany, something very near and dear to her heart.
Hi, I'm Justine McKnight, and I'm an archaeobotanist. Archaeobotany is the study of plant remains preserved within archaeological sites. And today we're going to talk about how we do archaeobotany and what it teaches us about the past. We study archaeobotany to understand how people in the past used plants to fulfill a variety of needs, from food to clothing to architectural materials to medicine, and even the use of plants in things like spirituality and religion. At the same time, looking at archaeobotany helps us to understand the ways in which people, in the course of collecting or producing plants and plant products, how people affected plant species and how people affected the environment in which those plants grow. So it was a pretty symbiotic relationship in a lot of ways, and archaeobotany helps us to gain a better understanding of that process in the past. Here in Maryland, most of the archaeobotanical remains that we get to study have been preserved through carbonization or burning. Occasionally, some kinds of contexts like privies or wells allow for plant remains to be preserved unburned or fresh in anaerobic conditions.
How we get that information is by studying plant remains that are visible to the naked eye, things like seeds and nuts and wood charcoal, and the remains of farm products like corn, beans, and squash.
And one of the ways we collect those plant materials is through a process called flotation, using a flotation machine, and that's what we're going to take a closer look at today. So we call this process flotation because we are encouraging floating organic material to separate from sinking heavier material. Essentially, this machine is a big vat of water. It has two sides. One side we introduce the soil sample into the sample basket. When we turn the machine on, jets of water circulate below the sample basket, and air jets agitate the sample gently to encourage floating organic material to rise to the surface and to overflow into our capture screen. So here we are preparing the sample. The flotation sample is composed of dry, unscreened soil collected from an archaeological feature. And we need to emphasize the importance of recording. We need to record the provenience, where that sample came from, and we need to record the size of the soil sample, usually by measuring the volume of the soil before we process it. By knowing the volume of the soil sample, that will help us to make statistical calculations about what we find, and that will make the data more meaningful. So now our sample is ready to introduce into the flotation tank.
Justine puts a clean capture cloth into the capture screen to collect floating organic material before pouring the soil sample into the sample basket.
So here in the light fraction screen, we see a variety of carbonized plant materials that are pretty recognizable to our naked eye. We see a corn cob, we see an acorn here, and wood charcoal, as well as a lot of other carbonized material that we're going to get a closer look at when we take the sample inside and take a look at it under the microscope. We're going to gather the light fraction sample up and put it aside to dry in a shady environment. So before we remove the heavy fraction, we're going to put a clean capture cloth down. We're going to lift the heavy fraction basket out of the flotation tank and flip it over on this handy work surface. We're going to use our hose to clean the heavy fraction from the sample basket. It's going to be collected here in the capture screen. And then we're going to gather up the heavy fraction sample, put it with the light fraction, and set these samples aside to dry. So now we're in the lab with our dried products of flotation, a heavy fraction and a light fraction. These two samples are from Montgomery Park's Josiah Henson site, which is a late 18th century feature here that we're going to take a look at today. So the first step in our process is going to be to put each of the fractions through a series of geological sieves that will just facilitate our process of analysis by allowing us to look at things sort of by size class. After that, we'll begin sorting the plant remains from the matrix, either the stone, gravel, the geologic matrix of our heavy fraction, or our light fraction matrix, which often includes things like small roots, carbon dust, or sediment that got suspended in our light fraction. So once we've separated our carbonized plant material from the flotation matrix, we sort it using a dissecting microscope into classes of plant material. Divisions like wood charcoal or nutshell or seeds or the remains of farm products like beans or corn. Following that, we proceed with a taxonomic identification of those plant materials. So we want to know what kinds of seeds are in our sample and what species of wood is represented in our sample. The tools that we use to do that are published keys available in books and on a variety of websites, and a reference collection of plant materials that we've collected from the geographic area in which we're working. So my reference collection includes things like nuts that are endemic to our forest here on the coastal plain of Maryland, if that's the site that I'm working on. The reference collection also includes nuts in their natural unburned state, but I've also carbonized nutshells so that when I'm making a comparative identification with archaeological nuts, I'm looking at burned modern nuts from my reference collection, as when things are carbonized, they undergo some shrinkage or distortion sometimes. The data that we collect includes a raw count of the kinds of specimens represented in our sample and aggregate weights of different kinds of plant materials, and that allows us to make valuable comparisons about our sample with other data sets. So archaeology relies on the expertise of a variety of different kinds of specialists, people who look at ceramics, folks who study lithic debris and projectile points, folks who are experts on buttons. Archaeobotany lets us delve deep into the relationship between people and plants, and in my opinion, that's a really essential relationship in the human experience. We eat plants, we build with them, we use them for medicine, and when we get the opportunity to look at an archaeobotanical assemblage from a site, we get a window into the way people who lived and worked in that place interacted with the natural landscape and how they modified it to meet their needs. So I think it's a really important piece of the human experience, and it's been a joy to talk with you all about it today.